Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at the mRNA vaccine clinical trial data from Pfizer and Moderna. So hang around with me throughout this entire video to get all of the relevant background information, so that way we can dive into some exciting experimental results. Plus, if you look in the description below, there is more information and instructions on how to get a free NFT. Now, before we get into talking about the vaccines, we need to set the stage. COVID-19 is the disease caused by SARS-CoV-2, and it was declared a pandemic by the World Health Organization on March 11, 2020, and has so far caused over 257 million cases worldwide and has been a contributor in over 5.1 million deaths. The consequences of the pandemic are far-reaching. This event has impacted environmental, social, economic, governance, and health systems around the world, and has been felt by just about everybody personally in one way or another. Symptomatic COVID-19 infections usually result in a cough, tiredness, fever, and loss of taste or smell. However, there can be more severe symptoms that can result in chest pain, loss of speech, mobility, or cognitive function, and can result in a difficulty breathing. SARS-CoV-2 is a coronavirus. It is made up of many proteins that allow it to survive in its environment and to enter into and infect host cells. SARS-CoV-2 infects host cells by binding the human ACE2 receptor with its spike protein. Upon binding, the spike protein gets cleaved, allowing it to initiate fusion with the host membrane, allowing the entry of the virus. Vaccines are an important public health measure that can be used to fight infections and alleviate the burden of disease on people and populations. Now, vaccines are not a new topic. The first recorded evidence of them date back to the 16th century in China against smallpox, while the first scientifically documented use of vaccines was by Edward Jenner in 1796. Since then, we have developed many vaccines against many different types of pathogens, which include viruses and bacteria. There are many ways that we can induce this immunity. We can use weakened or inactivated pathogens, or even just parts of them, to train our immune system to better protect us in the possibility of encountering that pathogen throughout our lifetimes. Some common vaccines are those against influenza, chickenpox, measles, and polio. These vaccines have variable effectiveness depending on the pathogen they are targeting, but do help to keep those who take them safe. Through the use of worldwide vaccination strategies, we can actually rid the planet of diseases. We've done so in one case, in the case of smallpox, and are very close to being able to eradicate polio. Now, because vaccines help to train our body to fight diseases, there can sometimes be transient side effects. These side effects can occur from any type of vaccine, like from the flu shot. These side effects can include fatigue, pain and swelling, muscle aches, headaches, and low-grade fever. However, they usually dissipate within a few days of receiving the shot. Now, a newer type of vaccine uses parts of the genetic code to help protect us from pathogens. They can utilize non-harmful virus vectors, messenger RNA, and DNA. Now, mRNA vaccines, and in particular the ones that are unable to self-replicate inside of host cells, like the Pfizer and Moderna ones, so they get cleared after they have created their proteins, have some really promising uses to them. Just to be clear, mRNA and DNA are two different things. DNA contains genetic information in it and is double-stranded. RNA is a copy of that information that goes out into the cell to facilitate production of the encoded proteins. Put it another way, DNA is like the blueprint of something that you have on your computer. RNA, and in particular messenger RNA, mRNA, is like a printed blueprint of what you are going to build. What you build is the proteins that do different functions in the cells. Now, if they are foreign proteins, like those of a virus, they can be sent to the outside of the cell, whereby it can interact with other cells around it, and in the case of the coronavirus spike protein, it can train the immune responses against it to better protect against SARS-CoV-2 viruses if they enter your body. These mRNA vaccines use a delivery mechanism called lipid nanoparticles to be able to get into the cells and utilize the pre-cleaved form of the spike protein to train the immune system. Just like vaccines have been around for a long time, so too have the individual pieces of the technology used to create mRNA vaccines. It first kicked off with the isolation of mRNA in 1964. The first mRNA vaccine efficacy study was performed in 1995 in mice, and the first human clinical trial was in 2008. Talking about clinical trials, they are a crucial final piece to developing medicines. Clinical trials are composed of 3 different phases to ensure the safety of the volunteers. When creating vaccines, their product is first carefully inspected to ensure reproducibility and quality across batches. These vaccines are then first tested in small groups of people, which help to determine if the vaccine is safe, if it works, if there are any serious side effects from taking it, and what the optimal dosing is. So that's in phase 1. In phase 2, the size of the volunteer population increases and the researchers ask what the most common side effects of the vaccine are and how the volunteer's immune system responds to the vaccine. Now, in phase 3, this is where the population size increases to hundreds to thousands of volunteers. This phase tests what the most common side effects are and whether the vaccine is safe and effective by comparing against a control population of people taking a placebo. Now, if the vaccine is proven to be safe and effective and the benefits outweigh the risks, then the FDA licenses the vaccine for use by the general public. I want to take a moment and really highlight why this type of research is important. It's important because COVID has directly affected hundreds of millions of people and has influenced billions in a worldwide pandemic. And vaccination is a way of preventing symptomatic disease. To help end the pandemic, it is imperative to test, implement, and approve public health strategies that will lessen the burden of disease on the population. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. Now, this brings us to one of the papers that we're focusing on today. This paper is called Safety and Efficacy of the BNT162b2 mRNA COVID-19 Vaccine by Polack et al. From Pfizer. Now, in this paper, they looked at the phase 2/3 clinical trial data of their mRNA vaccine, which is given in 2 30 microgram shots over the course of 21 days. This study is an ongoing multinational placebo-controlled observer-blinded pivotal efficacy trial. And between July 27th, 2020 and November 14th, 2020, 44,820 people aged 16 or older were screened at 152 sites worldwide in the United States, Argentina, Brazil, South Africa, Germany, and Turkey. 43,448 of them received injections of either placebo or BNT162b2 vaccine, 21 days apart. At the cutoff date of October 9th, 37,706 participants had a median follow-up time of at least 2 months of safety data. The male to female ratio is almost 1-to-1, and the majority of participants were white, 35% of people were obese, 21% had pre-existing conditions, the median age was 52, and 42% of the participants were over 55. Now, a major question with phase 2/3 clinical trials are, is it safe? And what are the side effects? Overall, BNT162b2 patients reported more reactions than the placebo recipient. And of the 43,252 total participants, 27% reported adverse effects with the vaccine compared to 12% with the placebo, while 20% and 5% respectively were determined to be related to the shot. Now, this distribution largely reflects the inclusion of transient reactogenicity events, and the distribution of severe and life-threatening events were similar between the vaccine and placebo groups. Now, on that note, few participants in either group had serious adverse reactions or adverse events that led to withdrawal from the trial. When closely following the reactogenicity events in 8,183 participants, overall, BNT162b2 patients reported more reactions than placebo recipients. When looking at the local effects in the first and second doses after 7 days in younger populations, the most commonly reported effect was mild to moderate pain at the injection site, with less than 1% reporting severe pain. And this finding was reported less in older populations. And these local reactions typically resolved in about 1 to 2 days. Now, when looking at the systemic reactions around the body, these findings were more often reported by younger populations, and more often after the second dose was administered. The most commonly reported symptoms was fatigue and headache, and the frequency of any other severe systemic events after any dose was less than 2%. Now, these data trends were similar, but less drastic in older populations. In general, Older populations were less likely to use antipyretic medication, while the placebo group was even less likely. These systemic effects were observed within the first 1 to 2 days after vaccination and resolved shortly thereafter. Now, another important question about this vaccine is how effective is it at preventing infection? When looking at the whole population, whether or not they had previous SARS-CoV-2 infection, the authors found that BNT162b2 vaccine had a 94.6% efficacy at preventing symptomatic infection 7 days after administering the second dose. While excluding those who had previous infections, this efficacy rose to 95%. Between the first dose and the second dose, 39 cases in the BNT162b2 group and 82 cases in the placebo group were observed, resulting in a vaccine efficacy of 52% during this interval and indicating early protection by the vaccine, which started as soon as 12 days after the first dose. This is what the cumulative incidence looks like when comparing the regular and severe cases, which are highlighted, over time. Of the 10 severe cases of COVID in this trial, only 1 occurred in the vaccine group, suggesting that the vaccine helps to prevent severe disease. Supplemental analyses indicate that vaccine efficacy among subgroups defined by age, sex, race, ethnicity, obesity, and presence of coexisting conditions was generally consistent, around 95%, with that observed in the general population. These are some pretty promising findings from the first approved mRNA vaccine. Now we will look at the second approved mRNA vaccine from Moderna. This paper is called Efficacy and Safety of the mRNA 1273 SARS-CoV-2 vaccine by Baden et al. From Moderna. And in this paper, they looked at the phase 3 clinical trial of their mRNA vaccine, which is given in 2 100 microgram shots over the course of 28 days. This study is a phase 3 randomized, observer-blinded, placebo-controlled trial performed in the United States. And between July 27, 2020 and October 23, 2020, 30,420 participants underwent randomization in a 1-to-1 ratio, where testing was performed at 99 centers across the United States. 15,210 of these participants were set to receive injections of either placebo or mRNA 1273 vaccine 28 days apart. At the cutoff date of November 25th, the participants had a median follow-up time of 64 days, and 61% of the participants had more than 56 days of follow-up. The male to female ratio was almost 1-to-1. The majority of participants were white, but had racial and ethnic population demographics generally representative of the US. The mean for this study was 51.4 years old, and 24.8% of participants were over the age of 65. In those under the age of 65, 16.7% of them had a pre-existing condition. Again, a major question with phase 3 clinical trials is, are they safe? And what are the dangerous side effects? Now, overall, around 23% of the people throughout the study reported serious adverse effects. The frequency of grade 3 medically attended and serious adverse events was similar between mRNA 1273 and the placebo. Only 8% and 4% of total events were attributed to the vaccine, with the more intense adverse events not topping more than 1% of the population. Overall, adverse events at the injection site occurred more frequently in the mRNA 1273 group than the placebo group for both the first and second doses. And adverse events were more commonly reported by younger participants, around 85% and 19% respectively for the mRNA 1273 and placebo groups. In the mRNA 1273 group, injection site events were mainly grade 1 or 2 in severity and lasted for an average of 3 days, so they weren't that bad. The most common event was pain after injection. Delayed injection site reactions after 8 days were noted in 244, 0.8%, and 68, 0.2%, after the first and second dose, respectively. This was characterized by redness, hardness, and tenderness, and resolved over the following 4 to 5 days after first reporting. Now, systemic reactions also occurred more frequently in the mRNA 1273 group after the first and second doses. And the severity of these reactions increased with the second dose, but still only lasted for about 3 days. In those that were positive for SARS-CoV-2 at baseline, these events were less common. When looking at adverse events over the course of 28 days, the frequency and severity was actually generally similar between groups, so it's just right at the beginning when these differences were noted. Looking further into the data, the authors of this paper asked, well, how effective is the vaccine at preventing infection? So when looking at the whole study population, the authors found that 14 days after completing both doses of vaccine administration, there was 185 symptomatic cases in the placebo and 11 symptomatic cases in the vaccine group, indicating a 94% vaccine efficacy. When expanding that analysis across the entire time range, 269 people in the placebo and 19 with the mRNA 1273 vaccine showed symptomatic infection. This resulted in a vaccine efficacy of 93%. And throughout this study, there were 30 people who developed cases of severe COVID, all in the placebo group, which resulted in 1 death. Now, when comparing efficacy across demographics, the efficacy was consistent around 94%. But older populations had slightly less vaccine efficacy, around 86%. Now, to quickly summarize everything altogether, in this video, we looked at 2 mRNA COVID-19 vaccines from Pfizer and Moderna. These treatments consisted of giving 2 doses, either 3 or 4 weeks apart, and were tested in over 30,000 participant populations. They found that both of these vaccines were around 95% effective at preventing symptomatic disease. Furthermore, both vaccines did induce side effects that usually faded after a few days and had very low incidences of serious adverse events. Now, not only do I think that these observations are exciting to investigate and learn about, they're also significant in a broader context. This information is significant because COVID-19 and the associated loss of life, health, and economic and social well-being can be prevented by immunization. These studies show a proof of concept for mRNA-based vaccines and are a promising new approach for protecting humans against infectious diseases, and ultimately illustrate the speed at which healthcare can move through coordinated efforts across society. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. Some of these questions are also informed by the limitations of the study. And I had a few questions myself after reading through these papers. Now, because both of these studies did not use sufficiently large enough population sizes to determine all of the possible effects of the vaccine, the question that I have is, what are all of the possible side effects? And on that note, what will happen over time? Now, because vaccination is meant to last for extended periods of time, and these treatments are still relatively new, this begs the question, will our immunity decrease over time? If that happens, will we need to take more shots to ensure that we are protected from this virus? This isn't a new concept. This happens with many vaccination schedules. Another question that was unanswered in these studies is what happens in the case of asymptomatic infections. Because vaccinated individuals can still get infected, just not show symptoms, and thus pass along the virus, how well do these vaccines help reduce that method of infection, if at all? As always, my final question focuses around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear your thoughts in the comment section below. Also, let me know if there are any topics that you'd like to hear about in the future. Now, ultimately, I hope that you learned something, but more importantly, I hope that you enjoyed your time doing so. So if you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I will see you next time.